Hello, my name is Gaia Mistriel, and my honors thesis studied the effect of testosterone on multidrug resistance protein 4 expression in a human kidney proximal tubular epithelial cell line. My experiment was conducted in the Department of Health Science at Eastern Connecticut State University, and my mentor was Dr. Amy Bataille. Have you heard of urate? Well, if not, urate is the metabolic breakdown of purines. Purines are organic compounds that are naturally found in the foods we eat and are found in DNA and RNA, for example, adenosine and guanine. And urate, or uric acid, is the result of the metabolic break and waste of purines. So urate acts as an essential antioxidant. It protects the body from free radicals. But eventually it needs to get excreted, and about 65 of it is excreted by the urinary system, and the rest is directed to the digestive system. So as you see here, we have purines, and then the breakdown, an example, adenosine and guanine, into uric acid, and then excreted through the kidney in urine. So the focus of my research was on the negative effects of urate. An excess of urate is known as hyperuricemia, which causes health issues. So the most common is gouty arthritis, which is characterized by the deposition of monosodium urate crystals. So basically they're crystals of urate in either the joints or soft tissues. So deposition in joints can be really painful. They can cause this acute inflammatory response that can wake patients up in their sleep. Other health conditions associated with hyperuricemia are kidney stones and chronic kidney disease, cardiovascular diseases, and metabolic syndrome. So when urate gets directed to the kidneys for excretion, it is filtered with the rest of the blood. Blood enters the kidneys and is processed in the functional units of the kidney, which are known as nephrons. And nephrons enter the blood first through the glomerulus, which is a small cluster of blood vessels. What enters the tubule is called the filtrate. Then as substances pass through the tubule, they may re-enter the bloodstream through adjacent peritubular capillaries, which are seen in blue. What is left in the filtrate after traveling through the entire tubule is what is excreted through the body known as urine, through this collecting duct. In the nephrons, urate is processed in 3 steps: filtration, reabsorption, and secretion. Urate is freely filtered by the glomerulus in step 1. And then in step 2, 90 to 97% of urate is reabsorbed back into the blood through adjacent peritubular capillaries. Next, tubular secretion occurs. This is step 3, when some of the urate is transported from the peritubular capillaries back into the proximal tubule. Finally, urate is excreted in the form of urine from the tubule in step 4. The cells that line the proximal tubule have a membrane that lines the peritubular capillaries known as the basolateral membrane, shown in red, shown near red here, and a membrane that lines the tubule called the apical membrane, shown near the yellow here. On these membranes are proteins that facilitate the transport of molecules across the membrane. So here to here, here to here. The multidrug resistance protein, or MRP4, is located on the apical membrane and acts as a unidirectional efflux pump for urate. So in other words, it transports urate from inside the proximal tubule cells out into the tubular lumen. And thus it contributes to urate excretion from the body. The function of MRP4 in kidney cells is proposed but not definitively proven. The ABCC4 gene, which codes for MRP4, has been associated with urate levels and gout in humans in genome-wide association studies. MRP4 also regulates urate in the liver, intestine, and blood cells. So we know MRP4 has a connection to urate and is a urate transporter. In avian renal proximal tubule kidney cells, it has been shown to be responsible for 75% of urate secretion. Other functional studies suggest that altering MRP4-mediated urate secretion could be a future target for hyperuricemic drugs. An important side note is that the mechanisms behind reabsorption and secretion of urate are still being understood. There still exists the question of whether reabsorption and secretion occur at the same time or one after the other. So that was step 2 and 3 in the previous slide. A better understanding of the transport proteins that line the tubule membrane and their role in urate secretion in the body can help in the development of drugs to treat hyperuricemia. What I've had found really interesting was that clinical studies have shown that hyperuricemia has a strong positive association with testosterone levels. So our first example is that men are about twice as likely as women to develop gouty arthritis. Second example, women diagnosed with polycystic ovary syndrome who have testosterone levels that are higher than normal have been shown to have a higher incidence of hyperuricemia compared to women without the condition. Individuals going through hormone therapy, like females transitioning to males, experienced higher likelihood of hyperuricemia with higher doses of testosterone. Then there's men undergoing androgen deprivation therapy, such as those being treated for prostate cancer, had significantly reduced serum urate levels corresponding with reduced testosterone. So when they underwent androgen deprivation therapy, they had decreased testosterone levels. Other studies have indicated that androgen receptors are present on kidney cells, so it is possible that testosterone could act directly on kidney cells to impact urate handling. Lastly, testosterone has shown to alter expression of 2 proteins that have a role in urate reabsorption, SMCT1, and that study was conducted in mice, and BCRP, which was conducted in a BWU cell line. So the objective of this study was to determine if testosterone affects secretory urate transporter protein, MRP4, in the kidney proximal tubule, and if that could be the reason why higher levels of testosterone are associated with hyperuricemia, so specifically studying the MRP4 protein. We hypothesized that the presence of testosterone would cause reduced expression of transporter proteins that promote urate secretion, MRP4, our example, when treated with testosterone. Reduced expression of MRP4 in humans could theoretically result in hyperuricemia. The cells used to test this hypothesis were cultured from the immortalized proximal tubular epithelial cell line, abbreviated as CIPTC. These modified cells express the essential transporters necessary for mimicking human renal reabsorption and secretion like MRP4, and you can see our cells here in this picture. So the first step in this experiment was culturing the cells, which took about a month, and then the cells were separated into different groups. The first group was untreated cells, previously tested for androgen receptors. The control group was treated with just DMSO, which serves as the vehicle for testosterone treatments. And then 3 groups were treated with different doses of testosterone for the same amount of time, referred to as the dose-dependent study. And then 3 groups were exposed to the same amount of testosterone for different lengths of time, and this was referred to as the time course study. So the next step was proteins were extracted from the cells to be analyzed. Proteins were separated based on size using SDS phage electrophoresis. This is when they were transferred from a gel to a membrane. And then the membrane containing the proteins was stained with a primary antibody, which tags MRP4. Secondary antibodies were used to bind to the primary antibodies, and this allowed visualization of the relative expression of proteins, which is known here as the Western blot. And this is our Western blot, and you can see our marker here. So this is one of the 3 Western blots after visualization was conducted using the MyBright imaging machine. And in the Western blot analysis, bands were identified at 150 kilodaltons as predicted for MRP4, and 42 kilodaltons as predicted for beta-actin, and beta-actin was used as our loading control. And no other nonspecific bands were detected as seen in this representative flow. As mentioned in our methods, the time course groups were exposed to the same amount of testosterone for varying amounts of times. They were exposed to 50 nanomolars of testosterone, and they were put into a 12-hour group, a 24-hour group, and a 36-hour group, and control. A one-way ANOVA statistical test revealed that there was a statistically significant difference in pixel density of bands between at least two time course groups, and this ended up being the 12-hour group and the control group when it was followed with the Tukey-Kramer comparisons test. The 24-hour and 36-hour were also greater than the control, but not statistically significant. The dose-dependent groups were exposed to different quantities of testosterone for 24 hours. So this included the control, the 50 nanomolar, the 150 nanomolar, and 450 nanomolar. Unfortunately, all the proteins from the 450 nanomolar group were not detected, and they were removed from data analysis. A one-way ANOVA revealed that there was statistically significant difference in pixel density of at least two varying amounts groups, and then followed up with the Tukey-Kramer again. It was determined that the band density of the 50 nanomolar group was statistically greater than the control, and also the 150 nanomolar was statistically greater than the control. Alright, so it was hypothesized in the kidney cells treated with testosterone would have less transporter proteins that excrete urate from the body. In this study, our hypothesis was proved incorrect. It was anticipated that MRP4 protein expression in any of the 6 testosterone treatment groups would be less than the control group, and in the present study, the opposite occurred. All 6 testosterone treatment groups had increased MRP4 expression compared to the control. So the cells in the testosterone low dosage, medium dosage, and short exposure had significantly greater expression of MRP4 than in the control group. So this was the first study to examine the effect of testosterone on MRP4 in this cell line. The relationship between MRP4 and testosterone may be more complicated than our hypothesis predicted because MRP4 exists outside of the kidneys. MRP4 can be found in the testes, for example. Additionally, a cell line can only capture a fraction of what occurs in a whole organism, and results from an in vitro experiment are not what would necessarily occur in the human body. You can't account for the mechanisms present in a whole body with just a single cell. Continually, this study used a small sample size. We had an N of 3 for each treatment group. Another limitation was focusing on just MRP4. There are many other proteins involved in urate excretion in the kidneys, and future studies may choose to test the effect of testosterone on other urate transporter proteins. These are my references for those 4 studies I referenced in the connection to testosterone slide. Thank you so much for watching and listening. This experiment was my honors thesis and could not have happened without educational and financial support from the honors program. Thank you to the honors program, and thank you to the professors that helped edit my proposal and final thesis, which includes Dr. Stish and Dr. Varapan. And the biggest thank you to my mentor, Dr. Bataille, for helping me design this experiment and teaching me along every step of the way and making my dream of conducting an experiment in a lab happen. Thank you to everyone.